Hey y'all, it's Mr. Reardon. It is Monday night, 10:25, and as promised, yet another mind-blowing screencast. This time, do something a little bit different. Rather than teach you some deep content, what I want to do is walk you through the mind map that does the overview for our Heredity unit and really show you how I've put these concepts together and hopefully show you how the larger concept relates to the smaller details. This lovely animal to my left, your right, is Lipstick, and her brother, Oscar. Not Oscar. Oscar's my dead cat. His brother, freaking, what's his name? Elroy is down here, and he may show up in just a minute. But basically, when I look at this Heredity unit, it's going to be quick. And I think about it as a wheel of really 5 concepts. I want to start with polygenic inheritance. We did that last week. Moving into some basic concepts about genetics, things you've seen before. And then our first real big new material, at least new for me and new for us. You've seen it before. I've taught it before, but I've never taught you guys this stuff. It's really understanding meiosis, and that is understanding how the recombination of alleles during meiosis contributes to genetic variability within a population. So much that I'm going to treat a Punnett square almost as a model, not just of one female, one male's offsprings, but a population of offspring. To do that, we're going to look at it 2 ways. We'll look at some basic principles of meiosis, things that should be real familiar to you since 9th grade. And then we're really going to dive into the more higher level concepts of meiosis on a second go round. And we'll look at meiosis basics as well as meiosis higher level with a model called chromosocs. Moving from there, we'll get into what the IB calls theoretical genetics, or what I'm going to call basic Mendelian genetics, kind of understanding how alleles relate to the things that Mendel saw back in the 1800s. Past that, we'll get into some more interesting stuff in terms of looking at dihybrid crosses and understanding linked genes. That is, why is it that someone who is fair-skinned and redheaded tends to inherit those characteristics together? We'll look at how certain genes are linked as opposed to opposite from each other. And then that'll kind of take us through it. I envision this unit going about 7 class sessions, and I am going to be really quick, and I'm going to make the assumption that you've had a lot of this stuff before. But without getting too far into it, looking at basic concepts, just a couple right here. We know that eukaryotic chromosomes are made of DNA and proteins, and we can definitely define gene, allele, and genome. Moving past that and getting into the basics of meiosis, here are things that you absolutely positively need to know. You need to know that meiosis is a reduction of division of a diploid nucleus to form haploid nuclei. You need to be able to define homologous chromosomes and outline the process of meiosis, going as far as to understand when homologous chromosomes pair up, when they cross over, and the fact that you've got 1 DNA replication followed by 2 nuclear divisions, and that's going to give us our 4 haploid cells. Moving a little further into meiosis, you want to know what happens when things go wrong, and that leads us to this idea of non-disjunction, which we'll discuss Tuesday or Thursday or Wednesday or Friday. Also, some real simple stuff. Know what a karyotype is. Know why karyotyping is performed, and then be able to analyze a human karyotype to determine gender and whether non-disjunction has occurred. Just as a little quick review/preview, did this morning with 4th block. That took us like 17 seconds. So that's the basics of meiosis. Let's look at some of the more high-level stuff. What does the IB want you to know? Well, absolutely want to be able to describe the behavior of chromosomes during the phases of meiosis. Want to look at crossing over and the chiasmata between 2 homologous chromosomes. Want to talk about how that crossing over results in exchange of genetic information, exchange of alleles, and then ultimately looking at how meiosis results in an infinite variety of potential offspring. We'll do the math on that in class, and then we'll get into Mendel's laws in particular, look at both the law of segregation and then here 2 assessment statements dealing with the law of independent assortment, which essentially deals with how chromosomes behave during metaphase of meiosis 1. All right, looking a little bit further down the line, want to get into some basics of Mendelian genetics. What you're going to see here are a whole lot of definitions. You guys will do this in your daily notebook, defining these terms here. And then, more importantly than just the terms, really understanding how to use the Punnett square to determine the genotypes and phenotypes of monohybrid crosses. And then we want to look at some more complex genetics, that is, look at multi-allelic traits like blood grouping. I want to specifically talk about ABO blood typings as an example of codominance. I want to talk about our sex chromosomes again and how they control gender, and then essentially be specific and understand that relationship between the X and the Y chromosome in humans, and then talk about how that relates to some human genetics in terms of knowing that the X chromosome is bigger, the Y chromosome is shorter, and then define sex linkage. And I think that we are actually more sophisticated than the IB. We'll talk about X linkage and talk about some examples there. Ultimately, moving forward, realize that human females can be homo or heterozygous with respect to X-linked genes. Understand that idea of having two X chromosomes can make you a carrier for certain recessive alleles. And then we want to be able to run Punnett squares and be able to run genetics problems on anything from monohybrid to dihybrid to X-linked to autosomal recessive to autosomal dominant. And then, of course, you can already do this, be able to analyze pedigrees and deduce genotype and phenotype. So a lot of terms, but nothing new. Again, use this map as a way to kind of keep your head straight and know that it's going to be okay. We know this stuff. We've seen a lot of it before, and we'll just keep on moving. This is going to be kind of new. I haven't taught this at the IB before, and that is understanding gene linkage. So here, playing with some dihybrid crosses, for instance, round versus wrinkled seeds, green versus yellow seeds, that stuff. We'll also have some more fun with it and look at things like color blindness versus dwarfism. That's a typical dihybrid cross that I like to talk about. Know your autosomes versus your sex chromosomes. We've already looked at that this year. Talk about the exchange of alleles again during meiosis, and then we need to look at how linkage happens, and that is the closer two genes are, or rather the closer two alleles are on a chromosome, the more likely they are to be linked together and travel together as a package during meiosis. And then, we'll come up with some examples of linkage, better ones than the ones that I just muttered through. And then we want to be able to figure out how to track the recombinants in a dihybrid cross. So again, more genetics problems, and we'll spend some time in class talking about that. And then finally, we'll end up with polygenic inheritance. You know it, you love it, you can't live without it. We'll define polygenic inheritance. We did that last Friday or Thursday, and then we will talk about how polygenic inheritance can contribute to continuous variation. This is huge. This is a huge concept because it really helps us understand how natural selection works on various traits within a population, and how that selection can ultimately lead to adaptation of a population over time. So you guys know me well enough to know I like to link everything together, but here, once you understand the basic genetics principles, you can really start to understand what it is that Charles Darwin and Alfred Wallace saw, and how it is that they came up with their theories for evolution by natural selection. You'll see these mind maps posted twice online, once as a folded up version, just the high line, basic concepts, etc., and then you'll see it unfolded, so you'll have a working schematic of all the concepts that we will look at over the next couple weeks. Look forward to working with you guys in class. See you at the IB.